In this talk, how to design using algorithm for epistasis test in human class. So the goal of the study is to find genetic locus that underline phenotypic difference. And the idea of Genome-Wide surface is basically the test for the association between the phenotype and the seeds that are spread across the touching. So I do process for GWAS. The first one is a bit of a database of genetic variations. The second one is high-level technology. So the first symbols the database of genetic variants. One particular project is HapMap project. And the goal was to identify and catalog genetic variations so that can guide the genetic study. And in the HapMap project, about 4 million genotypes. And in the ongoing project, in the next generation, we will have more and more genetic variants available. So the combination challenge is very difficult in the genome study. And this is my focus of this book, basically to tackle the computational issue that's associated with GWAS. And the genome data used in GWAS SNPs. So basically, the SNP is a mutation from in DNA sequence, and in Inbred mouse, in non-molecular conditions such as Inbred mouse, the SNPs are encoded as binary variables in 0 and 1. In human heterozygous populations, the SNPs are encoded as binary variables in 0, 1, and 2. And phenotype and binary, in case some studies of basically high individuals, we saw resolve disease by 0 and 1, or it can be quantitative, like a more size, things like that. And the best in GWAS is basically on different seeds that are associated with the phenotype that we are interested in. So, the two approaches in GWAS. First one is known as single-locus approach. In this approach, at each time we test the single and the phenotype. But many diseases such as heart disease and diabetes, they are known to be complex traits. That means they are highly dependent on multiple genes. So the single-locus approach, because it does not consider the interaction means it's not suffice to identify multiple genes that have interactions. And another class of work that is the single-locus approach is known as a two-locus approach. Also, it's also sometimes called epistasis detection. Basically, the difference is that in epistasis detection, each time is assessing the association between phenotypes. If we test the association between the phenotype and the SNP pairs, so we want to find which SNP pairs are highly associated with phenotype. Because we have so many SNPs and tests need to be performed in the whole genome, the main test so we need to care for the multiple problem. Basically, it says multiple tests increase the corner. Basically, if we perform one test with from 1.5 error rate, one error, then we have 64% probability to get at least one SNP result. So to control the error rate in GWAS, some ways of performing Genome is normally collecting this assumption that the test independent, which is certainly not true in GWAS because many of the SNPs are higher rate. So another method to control the error is known as mutation test. From this, basically we measure the phenotype values and reassign them to individuals, and then we test and use the distribution of to estimate the non-hypothesis distribution. So and the mutation test has been regarded the both and answer to assess the statistical significance of association test in GWAS, and the permutation test can be used to control both factor-wise error rate and both discovery rate. So these two methods are also in GWAS. So the challenges in epistasis detection, the background challenges. The first one comes from the statistical side. So basically, we have the difficulty is to develop effective statistical tests that can capture the interactions between genes. And there are many tests available, like NOVA test, G test, and many others that's still being proposed in the literature. So another challenge is coming from the computation side. Basically, it's almost search space. So for example, given 1,000 samples and 100 SNPs, say we just use 1,000 combinations, then the whole space for stasis detection is 5 times 10 to the power of 15. So this is a huge space. And here in this example, I only use 1,100 SNPs, and obviously it's a hard database, it's harder than that. So the computational challenge is really a crucial issue in GWAS, and this is what we want to tackle. Now let's examine some of the existing approaches to epistasis detection in GWAS. So the first one is exhaustive approach, basically you simulate all the events. And this one is certainly not feasible because there are too many of them to statistically enumerate. And so another approach is apply heuristics, like genetic algorithms. But this approach is not optimal because you apply heuristic and ignore some of the interactions of systems. And another actively used approach is not two-step approach. Basically, in the first step, you feel the needs to have a low negative effect, then you study interactions among the main needs. And because this approach is not optimal because it ignores the interactions between the seeds that have no negative effect. So another approach is use a space strategy from the genome search space for each in GWAS. So let's take a look at the recent development in this class of approaches. So there's some that's basically developed like fast ANOVA test, fast developed for bicycle test, and which is a general approach that can be applied to all complex tests and which is and some properties of this approach is that all use computations for error control, and the specific error control is optimized error rate. And the overall unpromising SNPs and previous method. For example, previous method only considered binary genotype, and the team can do postmortem number of target populations. And previous approach consists of one so in each case, number of samples is really small, and the team can do large sample study, and so basically it's specifically designed for human GWAS. And previous approach can only control finalized error rate and control standardized error rate and idea, which both widely in GWAS. And for the statistics that can be used, so previous approach defined the statistic for some properties, but T can be applied to any statistic that's based on a contingency table. So the computational problem is very straightforward. Given that SNPs x to x and phenotype y and its k permutations, the one k one would be the best value for all SNPs. That's a very straightforward formalization. And so we'll see later that the computing as a statistical C plus SE is almost equal to equivalent to computing contingency tables. And here is the contingency table for Two-Locus GWAS. So basically, each entry we record the occurrence of certain events. For example, the entry P2 prevent the occurrence of the event that ISJ and phenotype y they all equal to 1. And we've shown in the paper, because as you can see, there are 18 entries in this table. So we have in the paper shown that there are only 4 degrees of freedom. So basically, given the Two-Locus table and phenotype relationship between these, we only need to fill out the 4 entries in this table instead of all 8 entries. And there's a certain entry in this conversation script and instead of 4 entries to update, but the way we take is to the compilation cost. So next, I'll use this entry as an example to show you how we develop efficient algorithm to update contingency tables. So suppose we have a series the phi shown this in the figure on the left, and we want to find the frequency of events if I, J, and Y all equal to 1. So basically, we want to compute the curses of the event. Because we only have the event that when Y is equal to 1, so we can immediately from that part, so we don't have to consider samples with 0. And suppose now we already have, so you already know for skip x1, x2, the G2 value is 2, as these two samples are labeled by the red color, and the question is that how do we get the G2 value for skip x1, x3? So how do we do that? So as you can see, the x2 and x3 they only for 2 positions, and for the 2 samples that are labeled by red, there's only 1 difference. So immediately we know that the G2 value for snip pair x1 and x3 is 1. And so we don't have to send all the samples in order to get the G2 value for new snip pair, that is x1 and x3. So we can incrementally get the G2 value from previous snip pairs. And we can see the cost to update this entry is proportional to the difference between the two snips, the snip x1 and x3. And so we need to determine which one we use or update these contingency table. So that is to use the minimum span tree. Or if you're trying to talk about the minimum span tree, how we define it on the snips. So basically, for the snips, we can define graph, and the vertices in the graph can be the snips, and connect snips and use the difference between the snips as a weight of the edges. And for example, given 4 snips, we can draw the graph and label the weight between snips. And span tree basically is a tree that has all snips and different span tree without different tables. Let's take a look at these 3 things. So let's do the snip pairs associated with x1. So for the tree on the left, the total update cost for these 3 snip pairs is 9, is the weight of tree. And using the entry in the middle, the total cost is 8. And using the right, which is minimum span tree, total cost is 6. I mentioned the minimum span tree is 3 that have the minimum weight of all span trees. And so this is all very straightforward. It's a tree that has minimum weight guide the updating cost in order to reduce the cost or computational cost. And the overall idea of the algorithm is how to build the minimum span tree on the snips and a leaf in tree, which is also steep. We use a minimum span tree to guide the update process for snip pairs associated with x1. And after we complete the contingency problem for snip pairs associated with x1, we remove x1 and we repeat the same process for the snips in the remaining tree. So let me show you some. This is between the team algorithm and the using Q data. So we generate data from half time, which is oscillation, and the number of snips is 100,000, 400,000. Half of them are cases, and we just demonstrate the post-100 permutations. And the right shows the runtime of the post approach. The green segment here shows the right. So you can see the algorithm is about the post approach. And we also brought the run column of the algorithm. As you can see, the build line on bottom only takes the very small total runtime, and the most time consuming part is contingency table. So basically, that says to compute the test statistic basically is equivalent table. And the two expect to speed up the process and compute the company. So we also compared out with the previous method, GWAS, which is published in last history. This is the same for Inbred. And so compared it using Inbred data, and this is real data set, real data from a Jackson lab. And the number C in this example is 10,000, and the possible size is 1 on science, and the number per day is 100, and the K rate is 1. So X here represents the number of individuals in the set as we can. So red here, the top line, red line, the proof approach to give the reference. And in the middle is the time of so which is 6 grams, and the red line here is the team algorithm. And as we can see, when the number of individuals are large is less than 50, then the team algorithm become much more efficient than the proceed algorithm, even for Inbred data. And this shows the pruning ratio of the team algorithm using Q data dimension. So basically, the first column shows number of snips using the experiment. Second column shows the pruning ratio when we use minimum span tree to guide the update. So the pruning ratio is defined as the updating cost of the error divided by the total number of samples. So as we can see, we also some trees to guide the search the third column shows a real or random tree, and last one shows a real when using linear perspective, just like I skip here. And as you can see, using Genome-Wide tree, most of the samples are found without being scanned for Pruning table. And you look at how many samples are using Genome-Wide tree, there are only about 6% samples left in your scan. This one tree is about 11 samples in your scan. So basically, the form scan, so using Genome-Wide tree, we can actually be successful about 2 times. And summary, so we have the epistasis detection in human GWAS, develop equipment changes, and the idea of team is basically we use the incremental updating process to compute the table. For future work, we extend we focus on based on first study, and in the future we extend to constitutional types, which is very important. EQI study, and we also like to extend to material study because there may be multiple interactions among students. And finally, I'd like to think for the both for the PhD study, 2 NSF grants and 1 NIH grant for the ISMB program, travel fellowship in 7. Thank you. Okay, I got for you. Here is the CEO of Incremental. Sorry. Okay, so after the second one here, the time compares to second one itself, our basic display is for TI project. So as you can see, using both of here is what the rotation here, and number M is the number of individual case, number of permutations, and W is true weight. So the reality of K is shown in the second one here. So in the both approach, the time value K is instead of M. So and is dominant factor, and in the complexity of K, we move N, which originally is in from N squared to the last term here, which is associated with the tree. So basically the complexity of K. Okay. Yes. So BIM is popular evidence basic detection, and so one difference is that also is exclusive. So basically, it uses method to compute the error, and it is approximate. It uses any so it's not an exhaustive approach. So the error would be approximate instead of exact using computational-based method.
Maybe something, but I was wondering, could you comment on why do you need the DFS of this anyway, and not somewhere?
You mean benching?
Yes.
So basically you use the search then the you solve because this is a big trick. Although you use a search and there's a message you solve. That search then for all memory units. So this is a big constraint by the boundary size. And the overall efficiency is but using reference search will be much more.
Our question is the association analysis. If you don't, can we live with it? The second question is.
So with the question you mean the.
Friends means some other measures like obviously like age.
So this method is just the evasive method to compute the statistic value, so we don't take that consideration.
Okay. The second question is experimental K is about 100. In what value K be and where your algorithm can handle that?
So here we show the 100 page because we just show the performance. So for the performance trend will be the same if the samples will so 2 orders automatically faster compared to the root. And we have tried sort of implementation and run from in reasonable amount. And for the error of computation, we use I think the rate of 10,000. That's from my experience we are doing this time.
So there was more about missing human states.